Faisal, do you want
Yes,
to introduce your guest? Thank you.
sure. Patrick has a bachelor's in genetics from the University of Western Ontario, and he holds a PhD in biochemistry from Dalhousie University. With over 300 or more, actually, I lost count, publications in peer-reviewed, high-impact journals. Patrick is one of the most influential scientists today in the field of molecular evolution and cell biology of eukaryotes, especially protists. Patrick is the recipient of many prestigious awards, the latest of which was the 2021 Gilbert Smith Medal. And I've had the privilege in working with Patrick in his lab as grad student and a postdoc. His lab is one of the most active and dynamic labs I've seen and heard. And he's an excellent supervisor. He has a great eye to recognize talent. And from his lab, there are numerous people who have come out who are in their own place are very influential in our field. So without further ado, I pass it to Patrick.
Thank you, and that's very kind. Nice to see a couple familiar faces too. Hope all's well there. First, I'm just gonna, before I get started, I want to ask one technical question, which I can't see from here, which is, do you see my cursor moving around on this title slide right now?
Yes.
Okay, good. Well, I'll use that as the laser then. Okay, yeah, thanks again. I'll get started. I want to talk about some, I'm gonna mix up a little bit of new stuff, a bit of old stuff here, just because I want to try to unify a couple thoughts that have been bouncing around in different fields, bouncing around in my head, to how we think about the symbiotic relationships between microbes and their hosts. And these hosts can be microbes or animals. And so I'm going to try to bring a couple of threads together that we don't think about together too often. I want to stress maybe thinking a little bit about when we look at these relationships to think in a little bit more neutral terms and a little bit less about adaptation. Because when we see things in biology, we're always trying to assign a function. We see something and we go, oh, this must be for something. And I want to try to make the case that a lot of things we see in biology aren't actually driven by function. They're things that just happen and they're not harmful, so they are allowed to persist. And this is really true in a lot of interactions. It's true in molecular interactions between micro or macromolecules, but I believe it's also true in the interactions between organisms. So I showed this picture here because it's a famous case from archaeology of how context affects how we see function. So this was a crown that was discovered, and this painting is a picture of the crown of some long dead king. But when the archaeologists actually examined the artifact a little bit more carefully and thought more about its context and where it was found and how it was found, they realized that it was not a crown at all. I can't advance my slides, so I'll just do that. It's actually the top of a bucket, okay? They were just looking at it upside down. And so what is a utilitarian handle of a bucket was interpreted to be something ceremonial just because they didn't understand the context. I think that we do this a lot in biology where we're trying to see a function we believe is probably there and we sort of twist things around a bit to fit that. I'm going to give you a couple examples. First one, I want to use I'm going to do two different topics here. The first topic is about trying to get at the question of symbiosis and mutualism. So when you have a symbiotic relationship between two organisms, should we be assuming that they're both benefiting from it, as we frequently do? I mean, obviously, when you see parasites, you know they're parasites. And obviously, we know there's commensal relationships where one is thought to benefit and the other is thought to be sort of neutral. But by and large, people like to see mutualism when they see symbiosis. And so I'm going to use this organism here as an example. This is a ciliate called Eupodes. And it's a really interesting model system to study the interactions between hosts and symbionts. Specifically, I'm mostly going to talk about work that this postdoc, Vittorio Bosco, is doing in my lab And a lot of this is in collaboration with Claudio Benini in the University of Pisa, who's a really pioneer in the development of this model system. So Eupodes, as I said, is a ciliate, so it's a protist, it's a microorganism. This is its nucleus here. You can see this big C-shaped nucleus. Ciliates have a really interesting complex life cycle involving many different kinds of nuclei. I'm not going to talk about any today or any biology of ciliates because I'm just going to talk about their symbionts. So Eupodes is full of bacterial symbionts. All these red fibrous filamentous things are all bacterial cells that have been labeled here with fish. And if you look at the distribution of these in Eupodes in the phylogenetic tree, which is shown here, Eupodes is a diverse group with lots of clades. And this clade here, clade B, is the only one I'm going to talk about. Clade B is big, diverse, ancient, and every member of the clade has obligate bacterial symbionts living in cytoplasm. So, and they're all of the same genus, Polynucleobacter. So we infer that that symbiosis is ancient, so it took place right at the origin of clade B, and you can't separate the Polynucleobacter from the Eupodes. They're both mutually interdependent, so the Eupodes will die without the bacteria can be cultured without the Eupodes. So we assume that they're mutually interdependent. And this has all the hallmarks of a long-term symbiosis then. It's an ancient symbiosis that took place at the divergence of this big group, and all the members of the group still have the symbionts, okay? And this would largely be looked at as a mutualistic thing. It evolved a long time ago. They evolved to need each other and benefit from each other, and now they live together. That's all fairly straightforward. Polynucleobacter has been changed a lot by this relationship. On the one side here, you see free-living bacteria. They're really common in fresh water. They're just little dendroid things. But when they get inside Eupodes, the Polynucleobacter morphology has changed a lot. They become long thing in filamentous bacteria with these chains of nucleoids dotted through it, their cytoplasm. So the bacteria has changed a lot through this association. And what we would tend to see in this is like this, an ancient symbiosis. And this is something that's been studied really well in a lot of animals. There's a picture of an aphid here because that's the best model system. People like Nancy Moran have studied these for a long time and learned a lot about symbiosis. And the developed theory about these is that a long time ago, a symbiosis formed, in this case between this aphid and some bacteria. The bacterial genome shown here starts to reduce. Things are knocked out. Pseudogenes form. Pseudogenes are then lost. The genome gets smaller. At the end of the day, you have this tiny, compact, very efficient genome that doesn't have very many genes left, just the ones that it needs to survive and keep working with its host. And so with this fairly good model for how these kind of long-term symbioses evolve that has been really well worked out, again, by people like Nancy Moran. When you look at Polynucleobacter, you don't see this pattern at all. So we sequenced the genomes of many Polynucleobacters from different Eupodes. There are some free-living ones to compare them to. And what we see is this. So on one side over here, we have some free-living strains, just so you can get an idea what Polynucleobacter used to look like. And these are some symbiotic strains. And what we see is they haven't reduced to a small, compact, single kind of genome. There's actually quite a lot of variability. Some of them, like this one, is very similar to free-living one in its genome size and so forth. Other ones have reduced the numbers of genes in their genome size quite a bit. They're kind of all over the place. This shows how much loss they've had. So, you know, the biggest genome has actually lost a lot of genes. It doesn't have, this is the number of genes, and it doesn't have many genes. So there's a huge amount of variability. They haven't evolved to this tight little compact genome. And this is reinforced by the amount of pseudogenes and non-coding regions in them. So the free-living ones are all very similar to most bacteria in terms of the amount of non-coding that's coding, whereas the symbionts are all over the place. Some of them have almost half the genome as non-coding regions. Some are a quarter, but still more non-coding than you would expect to see in the strain. When you look at the actual genes, the chaos is actually even more obvious. You would expect that after a long period of evolution that they would have lost a bunch of now non-essential genes and pathways. But instead, what we see is just sort of a random pattern of gene loss across the different symbionts. So in each of these cases, this is a column is a symbiont, and these are just some pathways we picked. Green means they have the gene, and yellow means they lost it. And you can see there's no real pattern. So they haven't lost all the same pathways, and even in the pathways, they haven't lost all the same genes. It's just chaotic. It's not like a progression. The most important observation at all comes from phylogeny. So when you take all these symbiotic strains and you do a phylogenomic tree and compare them to how they relate to their free-living ancestors, ancient symbiosis would yield a case where you had a clade that was made up of symbionts because they all share a common ancestor, common symbiotic ancestor. But in the case of polynucleobacter, that's not what we saw at all. In fact, it was quite the opposite. So the yellow ones, orange ones are symbionts, the blue ones are free living. And if you construct the tree, you get a very strongly supported tree, the pattern of which shows that almost every single symbiotic clade arose independently from free living ancestors. Some of these are dead obvious, like here's an example here where we have a free living polynucleobacter very closely related to a symbiotic polynucleobacter. So there's no clade of symbionts and emerging from free living things. Every single symbiont has emerged independently from a free living ancestor. So what does that mean? How do you put that together? In a nutshell, you can say that this is an old symbiosis. It's an ancient symbiosis, but the symbionts themselves are not old or ancient. They're all new. So yes, I agree still that the symbiosis itself formed a long time ago at the origin of clade B euplodes, but in every modern clade B euplodes, you have a relatively brand new symbiont. So how can the symbiosis be old and the symbionts not be old? There's really one straightforward explanation. We go back to this idea that we have an ancient symbiosis, and then the bacterial genome evolves in a certain predictable way. The other implication of this is that the phylogeny of the bacteria and the phylogeny of the host will track each other because they've been co-evolving together. That's definitely not what's happening in this case of euplodes. Instead, what's happening is something like this, where we have recent symbionts and ancient symbiosis, and that's because they're constantly being replaced. So we have an ancient symbiosis that took place, shown in brown and yellow here, but it was quickly replaced by a new polynucleobacter at some point in time. And then as the euplodes have diverged and evolved, those symbionts have been replaced over and over and over again by a fresh influx of new free-living polynucleobacter. In other words, the polynucleobacter is picked up and it becomes an obligate symbiont within the euplodes. Its genome starts to degrade, it starts to lose genes and acquire pseudogenes, and the genome shrinks. But it doesn't evolve towards a stable, compact, long-term symbiosis type that we assume to be the outcome of such relationship. Instead, it keeps degrading. So the genomes aren't degrading in a sort of compacting, efficient way. They're basically falling apart because the host is making up for the shortcomings of the symbiont because the host needs it. And what this, again, Nancy Moran has theorized this would happen, and you go down this evolutionary rabbit hole where the genome spirals out of control, degrades, loses more and more genes, and eventually it becomes untenable. And so in this case, before that happens, the euplodes has simply replaced it. It's picked up a new polynucleobacter, which replaces the old one after its genome has sort of begun to spiral out of control. So you see a very different outcome in the symbiont genomes. In one case up here, you have the end. It's all fuzzy in mine. The endpoint is this tiny, compact genome, but there's an intermediate stage where it's starting to become pseudogenized. Don't confuse that with the case here, because these polynucleobacters aren't heading towards something. They're not heading towards this compact long-term stage, which this genome here represents, even though the genomes look fairly similar. This genome is heading for extinction. So once you're picked up, if you're polynucleobacter and you're picked up euplodes and you become a symbiont, your fate is sealed. You're dead. You're going to go extinct. This is not a beneficial long-term relationship. So you wonder, when you think about symbiosis, is this what we should be thinking about? I did a little search for crazy wedding photos. Oh my God, talk about rabbit holes. This is real, it's not made up. But this is kind of, when you say symbiosis, and then a lot of this comes back to the way it was pitched by proponents like Margulis, that it's this kind of happy union of two cells that's different from natural selection somehow. And, it's a whole new concept in evolution. I'm not really sure this is right. This isn't the right picture that we should have in our head when we're thinking about symbiosis. And I think it's more about conflict in a lot of cases than it is about cooperation. And so, a different example how you could picture symbiosis, and it might be something like this. And you don't see a picture like this, think about death row and think mutualism. But you could, because if you're a prisoner in death row, you're after by a host. The penal system is keeping you warm and dry and giving you accommodation. It's feeding you 3 times a day. And that kind of sounds like you're benefiting from that relationship. But in the long term, you're not. It's exploitation. And so if you think about the time scale, it's the same in these biological interactions. If you looked at polynucleobacter on a short time scale, you would say to yourself, well, the host is looking after the bacteria. It's giving sugar and carbohydrates and vitamins, and the bacteria is giving something back to euploides that we actually don't even know yet. But, it sounds like mutualism. And indeed, on the ecological time scale, you could see it that way. But on the evolutionary time scale, it's not mutualism, it's exploitation, simply because we now know that euploides is eventually going to replace that symbiont and it's going to go extinct. So euploides is in the driver's seat here. It's exploiting the bacteria for something. The bacteria is degrading in sort of slow motion over evolutionary time towards extinction. So this is not mutualism in terms of evolutionary time scales, although you could argue it's like mutualism in ecological time scale. So these contexts, like the time scale, is an important thing to get our heads around. We observe something in an ecological time frame and label it mutualism. We have to be very, very careful. So an evolutionary time scale, the picture might be extremely different. Okay, and I'm going to make this a little more complicated. It turns out that these euploides don't just have one symbiont. A lot of them have more than one symbiont. So here's a picture from Claudia, which shows the polynucleobacter I just talked about is green, but in red is another bacteria that's inside this euploides. Okay? So how do we deal with the second batch of symbionts? What are they like? Are they acting the same way as polynucleobacter, or is there something different going on? Turns out it's something completely different, and it's something that's probably really, really common. And that's because if you look at the evolution of these symbionts, they have another pattern again. It's not the long-term stable relationship that we saw in aphids. It's not the exploitive, relatively short-term replacement pattern that we see with euploides and polynucleobacter, but it's something else again. We call these accessory symbionts, but I also call them professional symbionts. And this explains why. So in this case, these symbionts are extremely ancient. Let's say back here, they've evolved to become endosymbionts of some hosts that we currently don't know. Their genomes did shrink and they're streamlined and efficient. They're not falling apart like a big mess like the polynucleobacter one, or like the long-term symbiont genomes. But they don't stay in one host. They don't form a long-term relationship with one clade and co-speciate with it. Really what they did is they just got really good at living inside other cells. And then they just move around to host species seemingly at will over evolutionary time. So they evolved to become symbionts, and then they moved into a different clade, and then they moved again, and they moved again, and they moved again. They move all over the place. So these are almost more like infectious agents, except that they don't cause any harm. Their brilliance is that they can get inside a new host, and they don't do any damage at all. They're completely concealed. But they're extremely efficient at getting inside host cells, living there without causing any harm, and then moving on in evolutionary time to a new host cell. So I'll give you a couple of examples of how common these are. If you look at every cultured strain of euploides from this clade, about half of them, they all have, polynucleobacter, the essential symbiont, that's shown in this first block here. But the ones that are colored in red, which is about half of them, also have this smattering of other symbionts. Turquoise means yes, brown means we haven't checked or no. And you can see there's none of them are particular. These are all the different kinds of symbionts up here. None of them is particularly common, but it's very common that Euplodes has at least one of them, and sometimes more than one, sometimes three of them living in its cytoplasm. And these are just from cultures. So it's occurred to us that cultures might not be a good representation either, because it's hard to say if these things would be maintained over decades of clonal culture, if they're not actually essential to the Euplodes, if they're not contributing anything to its survival, if they're just selfish. They're basically like, someone sleeping on your couch. It may be that once you get rid of them, you don't miss them. So Claudia just started this really great idea, I think, that we're collaborating with her on now, to take this back to natural samples at the cell level. So instead of establishing a clonal culture and believing that somehow represents the natural diversity, you go out and pick up single cells, use single cell genomics methods to see which symbionts are associated with that cell. And what we find in that case is nearly 100% of nearly 100% of Euplodes cells in nature have at least one, sometimes several, of these accessory symbionts. Here is a bunch of Euplodes cells on this left-hand side to the essential symbionts. I didn't talk about the fact that they can actually be replaced with a different genus from time to time, but you always have one or the other, or you die. The rest of these are all, some are very common, some are very uncommon, but there's a whole smattering of all sorts of these accessory symbionts that are associated with these cells. And these tend to go in a couple of clades of bacteria, which we're now referring to as professional symbionts. As I said, these are ancient clades of bacteria that long ago evolved to be really good at being endosymbionts of other cells. And because of the nature of the way we study bacterial diversity, they were originally discovered as pathogens of animals, and mostly. And so they were thought to be sort of pathogenic clades like rickettsia, stuff like that. But now that we're actually looking at them in nature in protists, what we're seeing is that the animal pathogens almost always evolved from benign protist bacterial endosymbionts. So these are clades of bacteria that have been, you can call it, infecting other eukaryotic cells for probably millions and millions of years. They mostly don't cause any harm. They do the same in animals, and once in a while, one of them is also pathogenic in it. Probably it's a recent transfer from some protist. So these are things like Holospiraceae and the Rickettsia in the alpha proteobacteria are good examples. These are basically not really, these groups shouldn't really be thought of as just pathogenic bacteria, but what they are is extremely well adapted specialists, professionals, if you will, that are good at getting inside other cells and living. That's really what defines these groups. And we're finding them now all over the place, hugely abundant, spreading between hosts rapidly. Probably the most abundant form of symbiosis in nature. Okay, so just to simplify this, just when I end this section, I want to break it up into two categories. Okay, and if you think about how these symbioses work from the direction of who's benefiting and who's driving it, it's actually fairly straightforward. You can have two types. You can have symbiont-driven relationships, like what I just talked about. This is the professionals. So they're the ones that benefit. They get inside their host. That's what they've adapted to do. That's how they need to survive. So they get inside a host and they live there for a million years or whatever. Move to a new host at some point. These are the pros. And so the symbiont drives the process. The symbiont benefits from the process. I think this is probably the most abundant form of endosymbiosis on the planet. These sort of fairly neutral reactions where the cell gets by, it doesn't do much harm, takes up a little bit of energy, but I don't think energy is actually, I think it's overrated as a currency. And so they don't really have much functional significance on their host. They can acquire functional significance when you have bacteria living there. That can't happen. That's not what drives the relationship. The flip side is the host-driven ones. So these are mostly things like what I described in euploides. I should say that that's not just euploides that's recycling its symbionts. The same thing is now being found all over the place with the well-studied animal symbioses. Things that we thought were long-term stable symbioses turn out to be cyclic exploitation and replacement events. Not all of them, but lots of them have been. So this is probably a pretty important form of symbiosis too, where the host picks up something that it needs, keeps it around for as long as it's still working, and then replaces it with something else and lets the old symbiont go extinct. And this is shown here with lots and lots of extinctions. And then once in a while, one of those relationships actually is stabilized from some change in the bacteria that we don't really understand. So this would be the case in the aphid that we used to think was the sort of model for how these symbioses work. But honestly, right now, it's starting to look like that's probably a minority of these systems. Those are special cases where somehow the host and the symbiont fall into a more stable relationship. But they're not that common. They don't represent most of the cases that we see in nature. So what about animals? Because obviously, symbiosis research really took off when people started getting excited about microbiomes. Microbiome research has changed a lot because people don't just think about bacterial pathogens anymore, but I would argue that it's almost as bad when we just start thinking about them in terms of the direct medical benefit to humans or physiological benefit to humans. But there's a lot of work on microbiomes that has changed how people see microbial diversity pretty tremendously. So there's lots of depth. The amount of data that's been collected is astonishing. There's more than a little bit of hype about the data, I'm sure. And what I want to talk about today is this third bit. There's not actually that much breadth in microbiome research in terms of kinds of hosts we're studying. Just to back that up, this couple of not very pretty graphs Earth Microbiome Report, and they just show sort of how many microbiome studies from different groups. Vastly biased toward vertebrates, of course, that's no surprise. A few plants, a couple of invertebrates. And if you dig into this invertebrates, which of course is a lot of animal diversity, see that they're biased as well. And so amongst the few invertebrate cases, they're hugely dominated by bees, flies, and aphids, which are all insects. They're all in arthropods, so they're all part of one phylum of animals. And collectively, then, if you look at the phylogeny of animal hosts shown here, we have all these phyla of animals, and virtually all our microbiome data comes from two of them, comes from a subset of two of them. Vertebrates are members of chordates, and insects are members of arthropods. So we don't have much breadth. We don't actually have microbiome data from a ton of different kinds of animals that's ever been sort of analyzed in a uniform kind of way. This is important because we're making a lot of assumptions about how microbiomes formed in the first place, like where they came from in animal evolution. And there's been arguments about whether most animals even have a microbiome as such, mostly coming from studies of Drosophila, where there's an argument that a lot of animals just have sort of a random subset of the bacteria from their natural environment associated with them, and not like a cohesive community of bacteria associated with them, which are very, very different, both evolutionarily and ecologically. So we started this project years ago. This is a massive project. Sounded great on paper, and then we realized how hard this was going to be, and I'm actually quite proud of the fact that we stuck with it and did this, because it was a lot of work. And what we did is we isolated more than 1,000 animals from 20 different phyla and characterized their microbiomes. So we deliberately targeted the tree as the distribution of diversity instead of picking a single group. A lot of microbiome studies would say, well, we want to do nematodes, and they do lots of nematodes. We devised a search strategy that would get us as much diversity as possible, with a little bit of repetition of the same kinds of animals too, but we wanted to spread it over the tree a lot, see what sort of patterns there were at large scale. And we picked different samples from different places, like different kind of habitats, mostly from around here. This was done with this Hackeye research network that supports our research. And so we picked their research stations, and we went to the tropics for a couple kinds of animals that we just don't have in British Columbia. And like I said, we had more than 1,000 of them. We built a very large image database and movie database of every one of the samples, which is actually a really great resource on its own because it's a massive diversity of animals. We enlisted a whole bunch of fauna taxonomists to help us identify them because we realized that barcoding is not an option here. Every single animal phylum needs its own barcoding strategy. We can't have 20 different barcoding strategies. We did the ribosomal RNA from the first few hundred, and it's not sufficient to identify them any better than an expert can looking at a picture. And so they're morphologically identified. Mostly, this worked very well. We didn't try to get down to the species level, but most of them obviously because these are understudied phyla. So then we sequenced their microbiomes as well. So we have over 1,000 microbiomes from different animals. What do we find? Here's some non-surprising stuff first, but good to know. First, they're less diverse than the environment that they're living in. So that's excellent. That's sort of what you expect. On the left here, we have microbiomes from 1,000 animals broken up by location or habitat. You can see the diversity index is around the same, and it's always much lower than the diversity index of the control samples, just of the seawater or the sediment or whatever it is that we've collected them from. So that's about 6, and these are less than 2. So it's a small diversity subset of the environment. That's what you would expect. Secondly, the microbiomes are distinct from the environment. That's really good news, and it's actually news because it could have been that they were just part of the environment. It could have just been a random subset of the environment, and it's not. So here in gray are the environment controls. You can see they cluster themselves in the different habitats, and the microbiomes, by and large, from the animal-associated communities, are different from the environments that they were collected from. So that's nice because it means they're distinct. And then this is probably most important. We find that the microbiomes share ASVs, or roughly equivalent to taxa. So we find that the microbiomes don't share many ASVs with their environment. So these diagrams, let's look at this one on the left first. The wedges are each a kind of animal, like a phylum, and the dots are a sample, like a community from that phylum. And the distance from the center to the outside is how little they share with their environment from where that animal is taken from. So if it was down in the center, it would suggest that the animal contained that 100% of the bacteria in that animal were also found outside of the animal in the environment. At the periphery, it would be 0. So the animal bacteria were all completely never found in the environment. And you can see that the proportion of bacteria in the animal that are in the environment is quite low, it's about 1/4. So most of the bacterial communities are not found in the environment outside the animal, which is very significant. And that's true of all the different kinds of locations and all the habitats. So this is the water column from the ocean. This is sediments in the ocean. About 1/4 of the bacteria that you find in any animal is found in the environment. A little more abstractly, but it's kind of interesting too. If you make a network of the environmental bacteria and how they interact with each other, the number of edges something has is how central it is to that network. So the amount of interactions that you tend to see between the bacteria in different environments can be quantified by its egocentrality. And if you take the bacteria that are very central to that network and you look at how commonly they appear in the animals, they're both low in abundance and low in prevalence. In other words, the most ecologically important bacteria tend not to be found in the animals in that environment, which again is just another way of saying the same thing, which is a very important conclusion, that these microbiomes in all these animals, well, not every single one of them, but collectively, are distinct communities. And so taking 1,000 tiny little animals, they do tend to have microbiomes. They don't just have a subset of the environmental bacteria. Like it's not just the food or whatever. But here's where we start to go off the rails a little bit from traditional microbiome thinking. We find that there's absolutely no link between the evolution of the animal and the evolution of its bacterial community. So here's just a clustering like what I showed of the bacterial communities in the animal different from the environment. But now we're looking at just the animals and classifying them by their phylum, and you see it's just chaos. There's no clustering of the bacterial communities by what kind of animal they're found in. And the phylum level is very high, but we see the same thing when you go down the different taxonomic ranks. It's not that we just picked a very high level. I just showed phylum as an example. I can show that in a couple different ways. If we jump down a little bit to the family, we had lots of annelids, we had lots of nematodes, so we were able to analyze those groups in more detail. And we also enlisted really good annelid and nematode taxonomists to help us more narrowly define what we had there. You can see in here, these are the different subgroups of annelids. There's no clustering by how they relate to each other either. So their communities are not similar in closely related annelids. And similar thing shown here, this is a heat map of sort of the similarity of the communities, and animals are ranked by their relationship to each other. So the closely related ones should be close to each other. So if closely related animals had similar communities, you'd see lighter, these sort of teal colored things around the axis in this heat map, and you don't. It's just random, it's chaos. And so there's no relationship between the phylogeny of the host and the kind of microbiome community that they have at this level either. In fact, you don't see this kind of relationship until you get to the species level. When you pick up the same species more than once, it is in fact more likely to have some overlap with members of the same species in the bacterial community. But it's not even perfect at the species level. There's a lot of variation between members of the species. There's very, very few taxa in the entire data set that are definitively attached to one species of host. There's overlap between species, so two closely related species can oftentimes share a lot of their microbiome. This is just a flatworm that we had lots of examples of and that we could identify down to the species level. So basically, it's all to say that there's very little evidence for any kind of phylosymbiosis. There's no relationship between community and the evolution of the host at most taxonomic ranks. It's something that's very short-lived. Maybe at the level of the species, a bit of it, but it evolves very quickly as the hosts emerge from each other. Okay, so then what are the communities then? If they're not specialized communities that co-evolve with their host over long periods of time, what are they? They're not just a subset of the environment, but not a long-term association with the host. So what are they? It seems to be that there's actually simply a grade or a guild, a guild of bacteria that are adapted to living with animals. And so this harkens back to what I was saying about euplodes, and I'll show you why. Remember I showed you this. So this showed that there is very few ASBs or taxa that are shared between the animals and the environment, right? That helps us understand that they're distinct communities. You do the same thing where you compare the ASVs in a host with other hosts from the same environment, you see a very different pattern. This is the environment, and this is other hosts. And instead of the communities clustering around the periphery of the circle, they gravitate into the center of the circle. And what that means is that the community of bacteria in an annelid from a particular environment tend to be very similar to the community of bacteria in a mollusk or something else from the same environment, not necessarily from a different environment, but from the same environment. In other words, there's bacteria out there that just want to be in an animal. So that's where they like to live. So the communities of bacteria in the animals in that environment tend to have more in common with each other than the environment itself, or between bacteria from other animals from different environments. And this is true, again, in the sediment, in the water, everywhere we live. So that's actually, I think, very, very interesting and important. So we have these communities that are distinct from the environment. They're guilds of animal-loving bacteria that want to get inside animals. And this really reminds me of the professional endosymbionts that I talked about, that are really good at getting inside the cells. And they just evolved to get inside cells, and then they move around through hosts in evolutionary time. These guilds are a really interesting clue maybe as to how microbiomes first evolved, but suggests a more of a bacteria-driven neutral kind of thing, where if animals tend to be home to bacteria that had evolved to be living inside animals, that's not really a functional adaptation from the animal's point of view. And it might have just led to these communities acquiring in some animals a more beneficial function to that animal just by virtue of the existence of the bacteria. But it wouldn't be how it first evolved. They might have first evolved as something driven by the bacteria, just as their favorite place to live. And only later did the animal, certain sets of animals, acquire some bad. It really asks the question, drove these associations between animals and bacteria that ultimately became the microbiomes that we're studying now. And I do think that to some extent we're studying microbiomes that don't really represent most animal microbiomes functionally, really at all. I'm going to end there, but I'm just going to say, because I'm a protostologist, I have to say that we also looked at the protists living with these animals. We didn't do all of them, but we did 300 of them. And we saw kind of similar things, but actually there's some fascinating differences. We saw guilds of things that tend to be, of protists that tend to be associated with animals, but interestingly, they're all guilds of protists that we already had called parasites, okay? And it really calls into questions whether these are actually parasites or just protists that like to live in animals that don't cause much harm. I think they're mostly commensal. There are things like apicomplexans that we call mouths, or these deep branching dinoflagella. They're really abundant. And it's also gratifying because those are exactly the kind of things you would have expected to find in animals, not just a subset of things from the environment, food. So that's ongoing work. There isn't too much eukaryotic microbiome work, so that'll be really interesting to dig our teeth into when we can finally get through these 1,000 bacterial microbiomes, which has been a lot of work. And with that, I'm going to thank the people. Corey and Vittorio especially led the whole microbiome project, which is funded by the Tula Foundation and the Hackeye Institute. And Corey's now leading the eukaryotic part, and Vittorio also did all the Euplodes work. So this is largely a talk of Vittorio's work. And then Emma and Philip are also our specialists on bacterial symbionts and protists, working in my lab and contributing in all sorts of ways as well. They work on these proto-symbionts that we find in lots of protists. And our work is also funded by the Moore Foundation. I want to thank Claudia and Brian for collaborating on the microbiome project and the Euplodes project. And that's all, if there's any time for questions.
Thank you very much, Patrick. It was great, very illuminating. I think lots of new things to think about. So if anybody has a question, please just go ahead, unmute yourself, and throw your questions at Patrick.
Professor Patrick, this is Vara. It is a really good talk about the microbiomes. I do have one question. May I proceed with my question?
Yes, please.
Yeah. During your talk about the host-symbiotic relationship, you said that the symbiont will lose some of the genes during that relationship. And so during the sequencing time, there is a chance of interference because of this gene loss to find the microbiome.
So let me just back up a little bit and say that when we did the microbiomes of the animals, we didn't sequence whole genomes. We just used the ribosomal RNAs, like the same as you would for any kind of environmental community sequence tagging. So we actually don't know what the genomes of those bacteria are like. We can make guesses based on where they go on the tree, but we don't know which ones of those are reduced and which ones aren't.
Yeah.
In the case of the bacteria living in the Euplodes, the ones that were completely sequenced were all things from culture. And so
Mhm.
we have like a snapshot of what that's
Okay.
like when we did sequencing. But
Mhm.
the reduction's ongoing. Like you said, if we went and did it 20 years from now, we'd probably see a different genome because it's probably lost more genes in the meantime.
Yeah.
And then the one the single cell things we haven't ramped that up to a genomic level yet. But that's what we'd like to do next. Does that
Okay.
answer your question?
Yep. Yeah. I mean, only thing is it should not interfere with the sequencing. That much of gene loss or gene gain in both of the side, I think, is not happened
Yeah,
until this 20 years of time.
that's right.
I
So it
think.
doesn't happen so fast that it messes with you
Okay.
while you're doing it. It's a slow enough process. We get really nice finished genomes where the um, the bacteria are all the same, which is probably because they're from clonal cultures.
Okay. Yeah. Thank you so much.
Are there any other questions?
I have a question.
Patrick, actually, I have a question. I was wondering, how you maybe reconcile between the neutral evolution, adaptive evolution within this context of microbiome, how they adapt, whether there is, as you mentioned, there is certain destination they are trying to reach, or a mix of both.
We have, this is almost getting into philosophy of biology because we've been trained all of our careers mostly to look at things and look for function and adaptation. But I mean, if you want to make the argument that you actually know that's the case, then you have to actually have measured selection. And we almost never measure the selective effect of a relationship between two things. So in the case of euploides, we know that there's some selection on euploides for keeping polynucleobacter in it, because if you treat it with drugs and kill polynucleobacter, euploides can't finish its life cycle. So that's very strong selection. That has been measured. But in the case of all those accessory symbionts, for example, there's only a very few cases. So Philip and Emma and Vittorio and me and colleague Julius Lukács and his lab just published this paper where we did this really long, comprehensive analysis of all the different bacterial symbionts who've been found with protists. And there's hundreds of them. And I think the number where the selection has actually been looked at between, like, if you were to knock out that bacteria, what's the effect on the host or whatever, is in the many hundreds of cases, maybe 2 people have actually looked at the selective effect of the bacteria on its host. And in those cases, it's always like, it's turned out to be kind of minimal. And we see this in the lab, although we don't measure it, it's anecdotal, where like some of Emma's cultures, where she's culturing these proteins called diplonemas, bacterial endosymbionts, and they were found that way in nature, and we've cultured them for a while, but every once in a while they lose them, and you don't even notice. The culture stays exactly the same. She only notices when she goes back and does fish, and then she's like, oh, the bacteria are all gone. So if there is a fitness effect on those, it's very minimal. And my guess is there isn't any, or insignificant, because I think that's the genius of these bacteria, is they can get inside a host and live there without doing any harm. So the host carries on fine, the bacteria is going to benefit from that. But it's that's just, yeah, it's more philosophy than science at this point because there's so little data. When you stop and think about it, it makes a lot of sense. Like most things in biology, when you actually think about them in a neutral, there's an argument to be made for lots of things that there is no adaptation, there is no benefit or harm. So it's just I guess it's just an idea to think about things in a different way.
Patrick, Jean-Francois. I was wondering if you guys check out these professionals kind of move from one organism to another. I'm kind of curious about the mechanistics of it.
Oh, so am I. I had this dream project to actually engineer movements, see what effect it has on the host. And Julius Lukács in Czech Republic, our collaborator who works with Emma, has actually we started doing that. Julius actually got it to work. He can take a diplonemid with one of these, it's a Microbiomes, like one of the alpha proteobacteria, and he can remove it and put it in a different diplonemid that has no symbionts and get it to stick, and it doesn't do anything. The culture is more or less exactly the same, which is sort of what you expect if they're just really good freeloaders. That's like they're like you're living on the couch. They're not doing too much. They're an annoyance, but not a huge annoyance, I guess. So I think this kind of thing, that's an anecdote, one case. But my thinking is if you did this with a whole lot of them, it's more just a matter of whether you find an environment that the bacteria is happy than it is whether the host can actually fight them off. They're good at flying under the radar. So there isn't, anyway, to answer your question, there's very little data on it, but it's starting to come, and it looks like you can actually transfer them.
Thanks.
Any other questions? If not, I want to thank Patrick again. Thank you so much for joining us. I know you have a very busy schedule.
Not lately.
Really? Good.
I just sit around in my basement like everybody else. But thanks for the invitation. I really appreciate it. I hope that made sense at least, or was kind of entertaining. Watching me flounder around with the slide and see what I can come up with. I hope it was okay.
It was great.
Yeah, it was fantastic. Thank you very much, Patrick, for joining us. Very enlightening presentation.
Thanks a lot, everybody.
Cheers.
Thanks for coming.
Yes, thank you.
Cheers.
Bye.
Bye-bye.
Thank you.
Thank you.